I got 271 over 300 in my A-level biology exam, and this is how I beat the examiner.
Okay, first thing is vehicle, right? There's 4 stages of respiration, being glycolysis link reaction, Krebs cycle, and oxidative phosphorylation. You need to be able to recall all those processes and know the only one which we don't form ATP is going to be your link reaction, which I've drawn out there. Cool. Now, they said ATP is broken down to glycolysis. My first thought is glycolysis. What's that? We look here. Glycolysis, we have glucose. It gets phosphorylated from fructose 1,6-bisphosphate. That activates it. So glucose was initially inactive. Now it's active. That breaks down. You form your 2GP molecules. You have reduction of NAD. You have phosphorylation of ATP, and you form your pyruvate. Yeah. So why is ATP broken down? Why is some ATP broken down? It's talking about the first step here. And the reason we're doing that is to phosphorylate the glucose so that it becomes active. And when it becomes active, we break it down to 2GP. Hope that makes sense to explain that question. Now, electron transport chain. Look, we could go through this long-winded explanation, but I want you to look at the diagram. Now, what do we know? We formed a lot of reduced NAD, a lot of reduced FAD through the reactions that preceded this. So what's going to happen is now it's going to get oxidized. Yeah. When it gets oxidized, we form NAD+, H+, and E-. Let's focus on the last two now. Your electrons are going to enter the electron transport chain, which I'm going to highlight temporarily. That's your electron transport chain. They enter it, and they're going to move through in a series of redox reactions. Yeah? Cool. When we do these reactions, we release energy. And that energy is going to be used to pump protons from the mitochondrial matrix into the intermembranous space. Yeah? Cool. That establishes an electrochemical gradient. Yeah? There's a difference in the charge, right? Electro, and there's a difference in the concentration of the chemicals as well. And we always want to move down our gradients to less charge, to less concentration. So we're going to move down the gradient through this ATP synthase channel, which looks a bit like a bottle of perfume. But we're moving through that ATP synthase, and then that is going to provide energy. The protons moving there provides energy for the phosphorylation of ADP to form ATP. And that is how we're going to form ATP. And that is the role of the carrier molecules in the electron transport chain. And you'll see that we've literally kind of said everything that I've written down here by using